use actually microscope to actually study the how the neuron cells handle the heavy metal copper and also the oxidative stress. So in our lab, it's actually trying to create some imaging approaches to really allow us to see the protein, a single protein one at a time in real time, so that we can actually really monitor how this protein work together. All the microscope in our lab is actually home built. So we need to actually purchase the individual parts and then combine those parts in a logical way so that they can actually do the things that we want it to do.
We also have the incubator to maintain the temperature and the humidity and carbon dioxide concentration.
In the lab, I mainly trying to develop some quantification method and use this one to study the protein behavior.
Because we're actually doing this real-time imaging, it allows us to treat the cell with different stimuli and see its response and how those responses induce a change of protein behavior. We can really pinpoint the amount of changes through our images. They will actually provide another layer of benefit to the scientific field to really understand how these proteins are actually working together to achieve the cellular functions.
Another new system we are trying to build is combining this super resolution microscope with the high resolved microscope. I think that one will be a very interesting system in the future.